appreciate it. So thank you so much for Dr. Greer for introducing me. So today, and also welcome everyone for attending today's departmental seminar. So today I'm happy to introduce my project, which investigates the role of two oncoproteins, TAZ and GRIP1, and their implications in breast cancer metastasis and therapy. Our lab focuses on breast cancer because it is the most diagnosed malignancy in women globally. Approximately 25% of diagnosed cancer in women are breast cancer, and it is estimated that 1 in 8 women will be diagnosed with breast cancer during their lifetime. Each year, over 600,000 cancer-related deaths are caused by breast cancer. But if you look at the survival rate in patients with only local tumor, it is roughly about 99%. However, it reduces dramatically when the tumor has metastasized to other places. In fact, over 90% of all cancer-related deaths are contributed by metastasis since there's no effective treatment options. So identifying new genes essential for breast cancer metastasis is critical for developing successful anti-metastatic therapy. And that leads to the focus of my project, including two genes encoding TAZ and GRIP1 and their implications in breast cancer. So starting with my first project, elucidating the role of TAZ in breast cancer metastasis and therapy. So TAZ is an oncoprotein regulated by the Hippo pathway containing several tumor suppressors. It functions through binding with a transcriptional factor tip in the nucleus to promote gene expression important for cell proliferation, cell death, and immune responses. Under
Can you just hit the OK blue button up there?
I don't have that in my screen right now.
Danny, can you
Sorry.
turn that off?
Sorry, what do you need me to do?
Oh, the recording button, like the OK for the recording.
I think you have.
I have to?
Be able to turn that off with your,
Let me see.
get your cursor up there and click on the OK button.
I don't have it. Like it doesn't
There it is.
it's like it missed. Yeah.
Yeah, you'll have to turn off the pointer function.
Okay. Let me see. No, I think it moves right through. It doesn't work that way. Let me see. Sorry about that. Where? Yeah, I don't have.
Turn your pointer off.
So it's turned off, but I don't see that.
So this screen is. Also, your Zoom, where you joined Zoom?
Yes, okay, so I'm seeing screen.
And you go to the presenter mode, maybe that'll.
It doesn't show me the,
Maybe just go back to just the PowerPoint.
like you want me to stop sharing?
Just like press escape and
There's no escape.
Stop sharing this. Share it again. Okay.
Okay,
Feel that or Danny turned off.
all right. Okay, let's.
Anyway, it's gone.
All right. Surprise. Okay.
Oh,
I'll
sorry, Annie, on Zoom, we've got your speaker notes up.
Sorry.
It's on like the presenter mode on our side.
Oh. Interesting. How do I? Okay, now it's acting weird. What happened? Okay. Oh my God. Okay. Share. No. Presenter. Oh, it freezed.
that's green, actually, it's outside the screen.
Yeah, okay.
You have to use filter.
Okay, let me just try that one more time.
Drag your screen into that. So that's happening. Yeah, drag your screen into this.
Let me, oh,
Back
sorry,
to
sorry,
the-
sorry, sorry. Sorry, I need to. But it freeze like as soon as I,
There you go.
okay. Okay, how about online? Also,
Actually, no, you're not sharing online, no.
okay,
You have to share.
okay, all right. How about now?
It's still on the presenter
Yeah.
mode. I don't know if you can swap displays, but that might change it to the.
I don't know how to, let me see. I'm not sure.
There is an option here, yeah, swap display.
No, no,
Now it's fine from our side.
yeah.
On your.
And then go to your PowerPoint.
Where's, okay. No,
And then you click, yeah, swap.
I don't see that. I don't see that on my screen. I don't,
Press present.
Yeah, using-
Sorry.
what?
Press present. On here, yeah. And
This one?
then what we can do is press cancel.
I don't see
You'll
it.
have to move your mouse, so
Oh,
it'll
it's
be
switched. I don't know why. I don't get it. Wait.
and then share screen down here. And then it'll ask you what desktop, and you want to choose that one.
This one?
Yep.
Okay.
And
This
then
sounds
it
good.
should show.
Okay. No, it's not
You
showing.
drag your screen into that, onto the right.
Okay.
Okay. Yeah, so now your screen is there.
Yeah.
Yeah, you just,
I don't see it.
yeah, you see it. Yeah, your arrow had to get into that because that is another place. Your arrow go to, right? Yeah.
Wait.
Now it's not good. Now it's good. Yeah, that's it.
Wait, now I cannot see my, I wanna, oh my God.
can't see it on your screen?
Yeah, no, I think it got switched for some reason. Wait, which three dots, please? Please. I don't, wait, which one?
This is your presentation you're
Yes,
sharing,
yeah,
right?
yeah.
Okay. Play from start.
But I wanted to.
You want to
use-
read your notes?
Yes,
Oh, okay. Use presenter view.
yes. Now it's.
Okay,
showing
and
orbit.
then now, yeah,
Display.
okay, yeah,
Swap display. Swap the display.
there we go.
Then they'll be.
And then the one you see on there is which one?
It's good on. Yeah, it's good.
Okay, then you're fine.
Yeah.
Didn't expect that to happen. Oh my goodness. Okay, all right. Okay, I'll. What a day.
We were just regulation of gene expression like that. You were done, Amy.
Okay. Oh, what a day. Okay, all right. So what else? Okay, all right. So starting over. So TAZ is an oncoprotein, which is regulated by the Hippo pathway, including several tumor suppressors. It normally functions in the nucleus to interact with a transcriptional factor in the nucleus to promote gene expression essential for cell proliferation, cell death, and immune responses. So under normal condition, TAZ activity is frequently inhibited by the upstream transducers. However, deregulation of this Hippo pathway is often observed in human cancers, causing TAZ hyperactivity, eventually leading to tumor formation. Indeed, in breast cancer, This abnormalities is frequently observed in breast cancer development. So overexpression in breast cancer is frequently observed in high invasive breast cancer tissues, and the correlation between patient survival and breast cancer TAS expression was also measured, even though the luminal and HER2 positive patient survival did not seem to correlate with the level of TAS. A lower survival rate in triple-negative breast cancer was detected in patients, especially with high TAS level. Since these patients also often exhibit a higher chance of developing metastatic disease, it indicates that TAS may correlate with breast cancer metastasis. However, there is still no such direct evidence proving that TAS can drive breast cancer metastasis. So that leads to the question of my first project: is TAS a driver of breast cancer metastasis? So to explore the role of TAS during breast cancer development, xenograft models were established by overexpressing DOX-inducible TAS as well as luciferase GFP in a non-tumorigenic mammary cells, NMUMG, which was injected into the nude mice. With the DOX treatment, tumor and lung metastasis were monitored. And by using fast sorting, we are able to establish two cell line tumorigenic and lung metastatic cells. And the metastatic phenotypes of each established cell line was then characterized both in vitro and in vivo. So to analyze the metastatic potential of these cells in vitro, functional assays were conducted. So the first one is the scratch-on assay to measure the collective cell movement. So after a scratch was made to the confluent cell monolayer, the collective cell migration was monitored by the cell imager over time. Another phenotype study is the cell invasion by transfer invasion assay, which uses the matrigel to mimic extracellular matrix. And in response to the chemoattractant added underneath the insert, the number of invaded cells that can pass through this matrigel will be measured by DAPI staining. And the second is the Anni-Ge assay to measure the anchorage independent survival. The cells will be plated into a low adhesive plate to prevent cell attachment. And after 3 days, the number of dead cells due to loss of cell adhesion was calculated using trypan blue. As for our in vivo model, each established cell line was injected into the nude mice, and the tumor formation and metastasis were monitored over time in these mice with or without DOX treatment. So starting from the in vitro study, first of all, the Western blot shows that no endogenous TAS was expressed in this cell line, whereas TAS overexpression was detected in these cells with the DOX treatment. Functional assays were conducted as described to measure the metastatic potential of these established cells. So I'll now, starting from the cell migration data measured by scratch-on assay. So as you can see in the orange bar, with no TAS expressed, low cell migration was observed in these cells, whereas TAS overexpression increased their ability to migrate, with the lung metastatic cells showed an even more dramatic impact with the TAS overexpression. When the cell migration was measured by the transfer invasion assay, as you can see here, low cell invasiveness was monitored in these cells with no TAS expressed, whereas TAS overexpression increased their ability to evade through the matrigel. And a similar increased effect was observed in the lung metastatic cells as well. And then when measuring the number of dead cells due to loss of cell adhesion by Anni-Ge assay, a particularly reduction in the number of cell deaths due to loss of cell attachments was monitored in cells with TAS overexpression, suggesting that TAS enhanced the cell anchorage independent survival, and this effect was even more dramatic in the lung metastatic cells. Overall, this data indicates that TAS did enhance the metastatic phenotypes of these breast cancer cells in vitro. But you may ask, how about in vivo? So the in vivo mass injection was performed as described before Re-injection of each cell line into these mice caused a TAZ-dependent tumor formation, as well as metastasis. And the lung metastatic cells exhibited even more dramatic metastatic events in these cells. So from there, we conclude that TAZ indeed drive both tumor formation and metastasis in breast cancer. Next, we wonder if TAZ is a potential therapeutic target for metastatic breast cancer. To address this, we sought to further determine two questions. One of them is if TAZ sustains the metastatic phenotypes of highly invasive breast cancer cells by genetic knockout using CRISPR system. And then the second being if TAZ can be targeted for therapy by identifying TAZ-specific inhibitors from a high-throughput screening. So now I'll show you the results for addressing the first question. So this is a Western blot showing the TAZ expression in MDA and B231 cells. As you can see here in the wild-type cells, high endogenous TAZ was expressed in these cells, whereas a loss of TAZ expression was achieved using CRISPR-Cas9 system. And then the functional assays were conducted to measure their metastatic potential. So I'll start from the cell migration assay measured by the scratch healing assay. So as you can see in the wild-type cells, they exhibited a high cell migration, whereas TAG knockout significantly reduced their cell migratory ability. Similarly, TAZ knockout also reduced their cell invasiveness as measured by the cell invasion assay. So as you can see here, the wild-type cells exhibited a higher number of cells that were able to evade through the matrigel, whereas the TAZ knockout cell had a significantly reduction in the number of cells calculated from the cell invasion assay. Since this is only one cell line, to avoid cell line specific effect, we also achieved the TAZ knockout in another highly invasive breast cancer cell line, BT549. As you can see here, with loss of TAZ expression, a reduce in both cell migration and cell invasion was observed in these cells, indicating that TAZ is indeed essential for maintaining the high metastatic phenotypes of these breast cancer cells. So overall, from our data, we conclude that yes, indeed, TAZ is important for the metastatic potential of highly invasive breast cancer cells, implying that it may be a therapeutic target for therapy. So we sought to identify TAZ-specific inhibitors from a high-throughput screening. But you might ask, how can we identify inhibitors targeting TAZ activity specifically? So if you recall my introduction part, TAZ is a transcriptional coregulator that works by interacting with a transcriptional factor TIT in the nucleus to regulate gene expression. So the protein-protein interaction between TAZ and TIT is thus essential for its function as an oncoprotein. But no TAZ-specific inhibitors have been identified yet. Therefore, we propose that disrupting such protein-protein interaction can inhibit TAZ both transactivating and tumorigenic functions. And this idea then leads to a collaborational screen with Dr. Fu at Emory University. So here is the entire workflow for identifying and validating TAZ-specific inhibitors. We actually established and validated several biosensors for monitoring TAZ interaction. I will now discuss the first assay used for the high throughput screening, which is the TR-FRET assay. So what is TR-FRET? TR-FRET stands for time resolved fluorescent energy transfer, and to produce the biosensor used for this assay, plasmids expressing either TAZ with a flag tag or TIT with a his tag were transfected into the cells, and the cell lysate were produced and combined with fluorophore or a donor or acceptor conjugated fluorophore antibodies against either flag or his, so that the TAZ or TIT protein can be detected using these antibodies. Upon protein-protein interaction, these antibodies will be brought close and the energy emitted from the donor because of the laser will be used to excite the acceptor, which emit light at a higher wavelengths. And both fluorescent signals can be captured by the omega microplane radar, eventually leading to a high TR-FRET signal. On the other hand, in the presence of different small molecules, any drugs that can cause disruption of TAZ-TIT interaction will eventually result a low TR-FRET signal. And the percentage of reduction by each drug wasn't calculated compared to no drug control. So by implementing this methodology, we are able to screen over 2,000 drugs, and then 22 FDA-approved drugs that were found to successfully inhibit TAZ-TIT interaction by at least 60%. So that's about 1% hit rate. So after we identified the initial 22 candidates, they were further validated by a primary assay using nanobit, which measures the TAZ-TIT interaction based on bioluminescence, giving us with 2 positive drugs, which was then subjected to a dual luciferase reporter functional assay measuring TAZ activity, eventually leading us with the final compound, Elucidating, which was also found to be able to disrupt the physical interaction between TAZ and TIT by several biochemical assays. And this drug can also inhibit the TAZ-dependent metastatic phenotypes measured by both cell migration and cell invasion. Because of the time, I will only show some results for the functional assays measuring both cell migration and cell invasion. So what is Elucidating? Elucidating is actually an FDA-approved antimicrobial drug, and it is found to successfully inhibit both TAZ-TIT interaction both in vitro and in vivo. To further examine its effect, highly invasive breast cancer cells with or without TAZ knockout was treated with this drug in different dosages, followed by functional assay to measure the impact on cell migration and cell invasion. So I'll start from the cell migration data measured by the scratch-owned assay. So in the untreated wild-type cells, you do see high cell migratory ability, which was inhibited by this drug in a dose-dependent manner. On the other hand, such inhibition was not observed in the MDA-MB231 cells with TAZ knockout, suggesting that this drug can inhibit TAZ-dependent cell migration. To avoid cell line-specific effect, we also used another cell line, BT549, and performed a similar experiment. So as you can see here, the wild-type untreated BT549 cell showed high cell migration ability, which was induced with Elucidating dose-dependently, whereas such inhibition was also not observed in the TAZ knockout cells. To further assess its anti-metastatic effect, we also conducted another type of functional assay measuring cell invasiveness. So here are the data for measuring the cell invasion by transwell assay. So very similarly, in the untreated cells, high cell invasiveness was observed in both wild-type cells, which was inhibited by increasing concentration of Elucidating. And such inhibition was also not observed in the TAZ knockout cells. So overall, this data suggests that Elucidating is a TAZ-specific inhibitor that suppresses the metastatic phenotypes of highly invasive breast cancer cells. So from there, we conclude that TAZ is indeed a driver of breast cancer metastasis, which may be targeted by Elucidating. And in the meantime, we also identified 6 additional drugs from another screen, which can degrade TAZ protein. But due to the time, unfortunately, I can't show them in depth. And next, I'll introduce my second project, which investigates the role of group 1 in breast cancer metastasis and therapy. But before I go into the details of my project, just some background information here. It is predicted that the human genome contains about 2% protein coding genes, while the rest is often regarded as junk DNA or dark matters. However, with the recent advances in technology, a novel type of protein coding genes was discovered within these dark matters, and they are called non-canonical open reading frames. So from now on, I'll refer them as ncORFs, just to save some time. It is well established that a conventional protein coding genes contains some regulatory regions upstream or downstream, as well as the conventional open reading frame. And transcription of a protein coding gene will give rise to a messenger RNA consisting of 5' and 3' untranslated region and the canonical open reading frame. However, recent study also revealed the presence of some ncORFs present within these untranslated region. But you might ask, what are the ncORFs? So basically, they are protein coding regions that were historically predicted to be non-coding based on some strict criteria. And the presence of these ncORF can also be found in long non-coding RNA, pre-microRNA, as well as circular RNA. However, despite the recognition of these ncORFs within the human genome, their biological functions are largely unknown. So it'll be interesting to further investigate their precise role in cancer biology, which leads to the focus of my second project, GRIP1. So what is GRIP1? GRIP1 is short for glycine-rich extracellular protein 1. And so far, there's only one paper published 4 years ago. And it is identified from an unbiased CRISPR functional screening. And it's found to encoded from an ncORF located within a long non-coding RNA. And it is also found to be a novel secreted protein. And this paper also links the importance of GRIP1 in breast cancer cell viability. However, its involvement in breast cancer remains mostly unknown. So to address this, we sought to investigate the precise role of GRIP1 in breast cancer progression. To study its role in breast cancer, we sought to ask 3 questions. So the first is, if GRIP1 can drive breast cancer tumor formation as well as metastasis, can GRIP1 be targeted for therapy? As well as how exactly does GRIP1 work during breast cancer progression? So now I'll show you the data for addressing the first question. Is GRIP1 a novel driver for breast cancer metastasis? So because there's no commercially available antibody, we first raised the anti-GRIP1 antibody from the rabbit to detect the endogenous expression across different breast cancer cell lines with distinct metastatic potential. So from this Western blot, as you can see, GRIP1 was not expressed in non-tumorigenic and breast cancer cells with low metastatic potential. However, it is preferentially expressed in highly metastatic cell lines. So to further investigate the role of GRIP1, we decided to overexpress GRIP1 in 2 non-tumorigenic cells, including NMUMG and MCF10A, which had no endogenous expression of GRIP1. So here are the results. So after the GRIP1 was overexpressed in NMUMG cells, a loss of ecoherent expression was observed, indicating a transition from epithelial to mesenchymal features, or EMT, which is a very common feature observed in metastatic cells. Also consistent with this data, the morphological changes was also observed after GRIP1 was overexpressed. So compared to the wild-type cells, which showed a more of a epithelial cell shape, GRIP1 expressing cells become more fibroblastic-like. Similarly, a transition from epithelial to mesenchymal was also observed in MCF10A cells after GRIP1 was overexpressed. To further confirm the observed EMT phenotypes functionally, different assays were conducted to measure their metastatic potential. So the same functional assays were conducted as described before. In this study, we also included an additional assay measuring single-cell movement by single-cell motility assay. So the single-cell movement was monitored by the same cell site imager every 20 minutes for a total of 24 hours. And the single-cell movement was measured by mean square displacement. Because of the time, I will only show you some of the functional data that I got. And here are the results measuring both collective and single-cell movement by both wound healing assay and single-cell motility assays. So as you can see here, so the wild-type MUMG cells displayed very little both cell migration and single-cell mobility, which can be increased dramatically with SCRIP1 or expression. And similar results were also observed with the human cell line MCF10A. So as you can see here, in the wild-type MCF10A cells, low cell migration and little single-cell motility was observed, whereas SCRIP1 or expression significantly increased its migratory ability, indicating that GRIP1 can enhance the cell migration. Moreover, since GRIP1 is a secreted protein, we are curious to know if purified GRIP1 protein can also increase the observed metastatic phenotypes in these cells. So we've purified the GRIP1 protein and then added them into the culture media of these wild-type non-tumorigenic cells. And very interestingly and consistent with the previous data, so the wild-type untreated UMG cell showed low cell movement, which can be increased by the purified GRIP1 in a dose-dependent manner. A similar effect by the purified GRIP1 was also observed using MCF10A cells. So low single-cell movement was observed in the wild-type MCF10A cell, and their single-cell mobility can be increased dramatically by GRIP1, purified GRIP1 in a concentration-dependent manner as well. Based on this data, we sought to further assess the effect of GRIP1 by another functional assay, which is by transwall invasion. So very encouragingly and consistent with the previous data, very little cell invasiveness was observed when these wild-type non-tumorigenic cells were not treated, but their invasiveness can be induced dose-dependently by the purified GRIP1. From there, we conclude that GRIP1 can enhance the metastatic phenotypes of non-tumorigenic cells in vitro. But how about in vivo? So we further ask if GRIP1 can drive tumor formation and metastasis in vivo. To answer that, we decided to use non-tumorigenic cells without or with GRIP1 expression and then inject them into the mice. The tumor formation and metastasis were monitored after the initial injection. So with the help of Rachel from Dr. Chris Nichols' lab, MUMG cells without or with GRIP1 or expression was injected into the mice individually, and the tumor formation was monitored over time. So here is a tumor formation result. So in the mice injected with only the wild-type MUMG cell, little or no tumor was developed over time. However, in the mice with the GRIP1 or expressing cells, you do see a large tumor formed, especially at week 6. And here's another way of showing the same result at week 6, which is the end of study. But more importantly, metastasis were also observed in these mice injected with GRIP1 or expressing cells, especially in the bone marrow and in the lung, indicating that GRIP1 indeed drive both tumor formation and metastasis in vivo. So from our data, we conclude that GRIP1 is indeed a driver for breast cancer metastasis, both in vitro and in vivo. So it is was investigating its therapeutic potential. So for this purpose, we specifically chose MDA and B231 cells, which is highly invasive and express a high endogenous level of group 1. So we decided to perform functional assays after inhibiting group 1 activity in these cells to assess the impact on the metastatic phenotypes. To inhibit group 1 activity, we first used anti-group 1 antibody since it is a secreted protein. And then functional analysis were conducted using MD and B231 cells after they are treated without or with these anti-group 1 antibody. So now starting from the cell migration data, so consistent with previous data, so in the untreated cells, high cell migration was observed, which can be decreased dramatically with the anti-group 1 antibody treatment in a dose-dependent manner. The single cell movement was also affected by this anti-group 1 antibody treatment. So consistent with the previous graph, very similarly, so high single cell movement was observed in the wild type cells, which can be suppressed dramatically by this anti-group 1 antibody treatment in a dose-dependent manner. We also performed 2 additional assays to further confirm this observed phenotype by both cell invasion and analysis assay. So it's very encouraging. So in the untreated cells, the cells exhibit both high cell invasiveness and a resistance to analysis, which can be inhibited by increasing concentration of antibody treatment. Overall, these results indicate that antagonizing group 1 activity could suppress the metastatic phenotypes of highly invasive breast cancer cells. To further confirm this observed effect, CRISPR was used to inhibit group 1 expression in these cells. And we also sought to include a CRISPR-resistant group 1 expression in these cells to see if it can rescue the observed phenotype. And functional assays were conducted as described before to assess their effect. So starting from the cell motility assay data, so similarly, high cell migration was observed in the wild type cells, whereas group 1 knockout significantly reduced their ability to their movement. But this reduced phenotype was rescued by adding back the CRISPR-resistant group 1 expression. Similar effect on both cell invasion and analysis resistance was also observed as well with both CRISPR knockout and add back of CRISPR-resistant group 1 expression. So overall, this data indicate that group 1 is indeed important for sustaining the metastatic phenotypes of highly invasive breast cancer cells. So from there, we conclude that inhibiting group 1 indeed impairs the metastatic phenotypes of highly invasive breast cancer cells, and it may be targeted for developing anti-metastatic therapy. So the importance of group 1 in breast cancer progression prompted us to further investigate its function. So in other words, how exactly does group 1 work during breast cancer progression? So what are the molecular mechanisms? We already know that group 1 is a secreted protein, which can increase the breast cancer metastasis as observed before. And this feature implies that group 1 may interact with some kind of receptor to promote its activity. So since group 1 sequences share a high resemblance to the extracellular matrix protein, we suspect that integrins may be the potential receptors. So after a deep dive of the group 1 sequence, the RGD motif was identified within the group 1 sequence. So we propose that group 1 may be a novel ligand for RGD binding integrins which are hydrodimers consisting of both alpha and beta subunits. To determine if it is true, we first tested that if this RGD motif in the group 1 sequence is important for its activity. To achieve this, the RGD motif within group 1 was mutated into triple alanine. The protein without or with this RGD mutation was then overexpressed in the non-tumorigenic cell line NMUMG, and the functional assays were conducted to measure the metastatic potential of these cells. So starting here's the result measuring the cell invasion. So as you can see, in wild-type cells with no group 1 expressed, low cell invasion was observed, but the cell invasiveness can be increased with group 1 or expression in these cells. But a complete disruption of the promoted cell invasion occurred when the RGD was mutated. To further validate the observed effect, we performed another functional assay measuring the single cell motility. So very similarly, when group 1 was not expressed in the cells, low cell movement was observed, but overexpression of group 1 increased their single cell movement significantly, which was destroyed after the RGD motif was mutated into triple alanine, suggesting that this motif is indeed essential for group 1 activity. But to further confirm if RGD enhances the metastatic phenotypes through the RGD binding integrins, we carried out additional experiments. One of them is by drug treatment using selumetinib, which can target several RGD binding integrins, including integrin alpha V beta 3 and integrin alpha 5 beta 1, which are already shown to promote breast cancer metastasis. So we then determine if inhibition of these RGD binding integrins by selumetinib will affect group 1 enhanced metastatic behaviors in these cells. So we first performed several functional assays, including the single cell motility assay. So it revealed that this drug, selumetinib, had no impact on the single cell migration of wild-type cells, but the mobility of group 1 or expressing cells was inhibited by this drug in a dose-dependent manner, indicating that group 1 enhanced metastatic phenotype depends on the RGD binding integrin. But to avoid potential non-specific inhibition by this drug, we decided to further confirm the importance of RGD binding integrins for group 1 activity using siRNA knockdown. So to avoid the non-specific inhibition, we decided to knock down the expression of both integrin alpha 5 and integrin alpha V by siRNAs in both wild-type and group 1 or expressing cells. After the protein knockdown, functional assay was conducted to measure the metastatic phenotypes of these cells. So this is the result measuring the cell migration by transwell assay. So as you can see here in the wild-type cells, despite the loss of integrin expression, there was no major effect on the cell migration, whereas a loss of group 1 promoted cell migration was observed after both integrin was knocked down in the group 1 or expressing cells. Overall, these data suggest that group 1 is an oncoprotein that may interact with RGD binding integrins to promote breast cancer metastatic phenotypes. After confirming the essential role of RGD binding integrins for group 1 induced phenotypes, we sought to further investigate the downstream signaling events. So because of the time, I cannot possibly show you all the data, but very briefly, by implementing similar method as described before, functional assays were conducted after inhibiting known downstream effectors by both drug inhibitors and siRNAs. And finally, we are able to figure out a novel GRIP1 mediated signaling event. So after GRIP1 binds to RGD binding integrin, three major mediators become activated, including LK, SRC, and FAC, and they then act on the downstream effectors, including TAZ and other mediators to initiate and promote the metastatic phenotypes, which then leads to breast cancer metastasis. So I know I introduced two projects. So just to draw the whole picture here, so we are able to confirm that GRIP1 is a driver of breast cancer metastasis, and inhibiting TAZ activity may be helpful for developing anti-metastatic therapy. Moreover, a novel RGD binding integrin ligand GRIP1 is identified to be a driver of breast cancer metastasis, which may also be targeted when designing anti-metastatic therapy. And just some future directions. Since we only did gain of function for our mice model, it will be interesting to look at how loss of GRIP1 expression will impact on the tumor formation and metastasis in vivo. So a loss of GRIP1 expression in MDA and B231 cell was achieved as described before, and they will be injected into the mice, and those mice will be treated with or without dogs to induce GRIP1 knockout and see how tumor and metastatic would develop in these mice. And also, we sought to generate and validate monoclonal antibodies and nanobodies against GRIP1 for both immunohistochemistry as well as for potential therapy. And also, once we have those specific monoclonal antibodies, the GRIP1 levels in actual patient samples can be measured both in the serum as well as the tissues to see if there's any correlation as seen in the breast cancer cell line. And also, further validation for the interaction between both integrin and GRIP1 will be carried out by different biochemical assays, including ELISA assay, and also to further investigate and validate the GRIP1 regulated downstream signaling event by RNA-seq, as well as so because of the time, I cannot possibly show you all the data, but we did perform an additional gain of functional screen using a library that we got from Broad Institute containing over 500 ncORFs, and we are able to find several candidates that were important for breast cancer tumor formation and metastasis. So I would like to take this time to thank my supervisor, Dr. Xiaolong Yang, for his constant help and supervision, and Dr. Greer and Dr. Andrew Craig for your support and guidance, as well as our collaborators, Dr. Jia, Dr. Chris Nico, and Rachel for the mice work, and also Dr. Fu at Emory University for performing the high throughput screening, and also special thanks for my young lab members. Thank you so much for their constant help, and yeah. So I'm now happy to take any questions.
Congratulations, that's an impressive story. I'm sure there's lots of questions. I'll open this to the floor then. And
Yes.
Dr. Gray.
Great talk.
Thank you.
Incredible results. Wondering for the last story, was this gene hard to find because it has like a non-AUG start codon? Or why was it missed before?
Yeah. So just to repeat the question for the online audience, Dr. Craig was saying if GRIP1 is difficult to find or miss because it doesn't contain a conventional start codon. So the answer is actually no. It actually contain a AUG codon for its translation. So I think it's because it's because of the location. and then the criteria of this initial screen when it actually taking place, when they try to identify potential coding genes, several like criteria were like the cutoff were made so that only the genes with very, very high confidence will be regarded as positive, whereas those with not so like high marks will be left out. So because of the presence, it's actually located in a non-coding RNA. So I guess that's why it got missed because it's located within a gene actually. So it actually located with actually I have the slide to show you here. So it actually it's located within a protein coding gene. So I think that's why it got missed.
So it's a
When
short
it's yes.
function.
Yeah, so that's why they were initially regarded as non-coding, if that answers your question.
Yeah.
Yeah.
Can I just follow up on that? So what's the other gene and is it in the 5' or 3'? How is it? How is it translated? Viruses, of course, you're a microbiologist from your early training. Viruses very often have multiple open reading frames,
Mhm.
sometimes overlapping different frames.
Yeah.
So this is in another gene. Is it completely separated from the open reading frame of that has been designated to be the open reading frame of that gene?
Uh, that's a great question, but just to repeat the question for the online audience and to see if I understand your question correctly, Dr. asked if this gene was missed because it's not the same open reading frame as the known as the originally identified protein coding genes. If that's
Well, sort of, it's just a second open reading frame within the same long transcript, or is there some other process by which it's spliced separately, or, you know, what's the mechanism by which it is segregated or differentially expressed relative to the gene that it seems to be sitting in?
mm-hmm. Oh, that's a great question. So, I think I think it's not in the same frame as the uh as the previously identified gene. So I believe it's a separate frame. So it's almost like a frame shift compared to the original protein coding gene. And I think that's why it originally got dismissed because it got included in a like a larger protein coding region. So after
Is it within the other open reading frame in a different frame? Or is it upstream or downstream?
that's a good question. I wish I could tell that, but there's so limited information on this gene. I don't think there's anyone that actually getting to the bottom of it. I wish I could answer you, but if I were to suspect, I think it's probably because it's translated within a different frame. So that's why, but it's like shorter compared to the original actual identified coding gene. So that's why it's like it's being missed.
Sorry to belabor this, but what is the other gene? Does it encode something interesting that might
Uh,
have functional?
not that I know of. Yeah. Sorry.
Dr. Nickel.
Yes.
Yeah, great work.
Thank you.
Just to follow those lines, just because of the table you presented, and it may be an unfair question given the limited information in group one. But given that it's what you've shown with grep one in breast,
Hmm.
when it's overexpressed. It's interesting that it's actually high in normal thymus, colon, and esophagus, but in all the cancer phenotypes, none of them relate to those normal tissues. So I'm wondering if there is a disruption in the tissues where cancer is expressed that is causing the shift in the translation within the larger gene, so that it's being overexpressed in those tissues and causing cancer where it normally might not be expressed.
That's a good question. And just to repeat the question for the online audience and to see if I understand the question correctly, Dr. Chris Nico saying if because in normal tissues it's usually high in the normal thymus and not the other ones? Is it because during cancer development there's a disruption for the other protein coding gene that causes high translation of GRIP1? If okay, so it's so I think from all the literature that I read, it could be a possibility. We do see that we do find a correlation between where is it? In the high metastatic cell line, we do see a high endogenous expression of GRIP1. Oh, but not the others. So we do suspect that GRIP1 may play a role, maybe non-causal or non-causal, for the transition between breast cancer duct carcinoma in situ to the invasive ductal carcinoma. So we do suspect that it may be a key, almost like a biomarker for them to become more invasive. So yeah. Yeah, we actually got another cell line. Yeah.
And one minor follow-up point, your last figure.
Mhm.
I understand why you maybe did it, but do you suspect there's a link between GREP1 and downstream TAZ signal?
Yes, yes. So just to repeat the question for the online audience, the question is asked if I suspect that there's almost like a crosstalk between GRIP1 regulated signaling and Hippo pathway, and I can say with confidence, yes. So actually, I don't have the time to show you them right now, but I did treatment with using the wild type UMG cell with at different time. You do see with the GRIP1, so and with the time you do see a inhibition of the Hippo pathway, but an increase in activity of TAZ. And this is also the case for the M7A cells. And we also did QRT-PCR. We do find a upregulation in mRNA level of CTGF and CRY61, which are known downstream target of TAZ. So it's very encouraging, and we are very happy to see these results. Yeah.
Dr. Lillicrap.
Yes.
Yeah, I mean, so first of all, congratulations.
Thank you.
On that humongous amount of work. That's really incredible. But as you were presenting, it was in the back of my mind that integrin signaling and as a non-canonical way of influencing YAP and TAZ was a possibility. And I was wanting to ask you whether you thought if you gave these two therapies, which you sort of come up with, do you get an additive or a synergistic effect, or so what would be your prediction if you gave cells or animals and you blocked both of these pathways? Do you think it would make a difference?
That's a very great question. So just to repeat the question for the online audience, Dr. Lillicrap asked if inhibition of GRIP1 mediated signaling and TAZ activity will cause a synergistic inhibition for the metastatic phenotypes. So I guess it could, but however, we do find that GRIP1 do mediate through other effectors, including rho RGTPases. So I would say there may be some energistic effect, but given the fact that we do suspect that GRIP1 activate other mediators, so inhibition, I think maybe almost like a triple inhibition may give you even more significant inhibition on the. But I do see, yeah, I do suspect there will be some.
Are you planning on doing comparative RNA-seq with the two different methodologies to see whether there are commonalities in the downstream signaling?
That's a great question. And to repeat the question for the online audience and to see if I answered the question correctly, Dr. Lillicrap asked if I'm going to perform two parallel RNA-seqs for both TAZ regulated and GRIP1 regulated signaling. So I guess it depends. And because previously we already did TAZ regulated downstream RNA-seq and we got the data already. So right now more, I guess more, that's more interesting. So we're more likely to focus on TAS regulated signaling pathways. So I guess,
Clarify this, you say you've looked at downstream TAS targets, but have you looked at them in the response to, say, adding GREB1 protein to cells?
yes,
Oh, okay.
Yeah. Yeah.
And you see the same targets getting regulated?
yes, yeah, yeah.
Awesome.
Yeah, so yeah, that's same again. So right now we're more likely to focus on group one regulated signaling, and so I think we are going to perform that this year. So hopefully, yeah. So hopefully we'll have some exciting results. For
So I have to ask this question
Yes.
because I'm supposed to be a developmental biologist of some sort, but not really. So does this gene exist in the mouse genome, and have people come across it? It sort of,
Mhm.
it really smacks of something that would be very important developmentally.
That's a good question. And just to repeat the question for the online audience, Dr. Greer asked if this gene is also share high homology in mice genome, if I understand it correctly. So the short answer is actually no.
It's not in the mouse gene.
Sorry?
You don't see it?
You don't, yeah, you don't actually, it shares poor similarity. I don't think there's even a group like a mouse group one protein in the mice.
Well, you didn't know about it in the human genome for an awful long time.
Yeah, but I guess, yeah, you're right. Yeah, I guess with
Well, just the same human gene that group one.
yeah,
is
so
part of.
that particular sequences share poor similarity with the mouse.
The gene that it sits in.
The
There is a mouse homolog of that?
no, I don't think so.
Okay.
Yeah.
Interesting.
Yeah.
Other questions? Oh.
This is just kind of a general question, but how does the GREB1 expression compare to the gene that is resting in? And it's like,
That's a great question. So just to repeat the question for the online audience, Tasha asked if so what's the correlation level between the gene expression and the group one, right? So the short answer is I wish I could tell you, but because there's so limited information on the like this group one, I'm actually not sure like what kind of gene that's located. I probably have to dig through the actual human sequence in order to find that. I wish I could tell you, but right now I don't have a definite answer, sorry. Yeah. But I would suspect that with increased expression, there will be a high chance of these translation transcription machinery occupying like in the nearby. So maybe it's like proportional, or maybe it's Cerveza and tiny natural. I'm not sure, but like just to give you some idea. Yeah.
And that would be something to iron out, because I think it's a kind of an interesting
Mhm.
aspect of
Yeah.
how this gene evolved.
Yeah.
And is it all coming, is it on the same transcript that's getting spliced, or there's differential splicing mechanisms going on? It would be intriguing if you could actually have ribosomes
Mm-hmm.
on this one transcript that are selecting that open reading frame to translate versus the other one.
Yeah.
So a lot of things, a lot of interesting questions. With that said, if there are no other questions, I don't see any other hands sticking up either online or in the room, so please join me in thanking.
Thank you.